which is the capillaries here, vasa recta. That is a term that we have to kind of dissect and figure out what's being exchanged here, although we have an idea, which is the solutes and the solution. Okay? Alright. So, we were well past this. Oh, we have the driving force here in this glomerular filtration. What's the driving force? There you go, blood pressure, absolutely right. And of course, this is also contributing now to the great filtration process that goes on. Very large afferent and small efferent arteriole. Okay? All right, and so we were kind of in this area, size and charge. Don't forget that question, that answer, size and charge. All right, that is something that's a question, I guarantee. Size and charge. This is how the filtration membrane selects for. Okay. All right. So we keep going here, and we were about right here, perhaps. Is this correct? We passed this. We're past that. Okay. So let's quickly do the granular cells. What do they produce? An enzyme known as renin, right? And what does renin do? Acts upon, yes. It cleaves angiotensinogen. And where do you, that's a plasma protein that is made by where, would you guess? Made by what? Do you have to guess anything? Guess the liver. Yes, it's made by the liver. Okay, so it travels in the blood. It's acted upon by renin and cleaved into angiotensin what? 1, yes. And then that travels in the capillaries, especially when it gets to the pulmonary capillaries, all right, because these endothelial cells are well endowed with what enzyme? ACE, yes, angiotensin converting enzyme, cleaving angiotensin 1 into angiotensin 2, which is think of this as a potent vasoconstrictor, and it is, okay? That's a good kind of nickname for that, and that certainly is. And so if it's a potent vasoconstrictor, what could we say that its main job is going to be doing? Trying to raise blood pressure, right? So that's one way we know that angiotensin 2 is going to act. It's causing vasoconstriction of the afferent and efferent arterioles, raising blood pressure. What's another way angiotensin 2 might try to raise blood pressure? We have a laundry list that we'll come to, but these are two that I mentioned already from the other day. It goes up to the brain, to the hypothalamus, right? Stimulating the thirst center, causing you to drink water, which increases blood volume and increases blood pressure. Okay? So we have angiotensin 2 potent vasoconstrictor, and now it also stimulates our thirst and hunger sensation, or more the thirst in that case. All right, be able to recognize what renin is acting on and what angiotensin converting enzyme is acting on too. Macula densa cells, what are they sensitive to? We know where they're at. They're the dense spot of cells right there, and so they're sensitive to the rate of delivery of the fluid and the saltiness, if you want. That's the word we can kind of use for the fluid here is saltiness. We can use that term instead of saying high osmolarity. Think of that as you kind of picture the fluid flowing through the tubes, saltiness, less salty, more salty. That's kind of a way of kind of keeping a track of the solutes, yes.
I put GFR, but I would be appropriate.
For what?
Glomerular filtration.
They are able to change the GFR. So they're sensitive to the amount of fluid that flows past them and the rate of fluid that flows, but they can do that by changing the GFR. So they say, hey, I'm sitting here, fluid's flowing past me, not very quickly, so I'm going to change the GFR, the glomerular filtration rate. I'm going to influence in the way that I can do that. And how do they do that? How do the macula densa cells do it? If the fluid is not flowing fast enough past them, they might release a what? Vasodilator, yes, known as nitric oxide, yes, acting on what? The afferent arteriole, causing it to relax, right? Vasodilate. And so now we increase the blood flow, increase the filtration, filtrate amount, now fluid flows faster through there. What if the fluid is not flowing as fast? I'm sorry, too fast would be the better question. Yeah. Too fast. Vasoconstrictor. And which one do they release? ADP. ADP is what they use for that. So potent vasoconstrictor in that case. So they're sensitive to the rate of fluid as it flows past them as they're sitting right there in the DCT. The last one is mesangial cells. What do they do? We know where they're at. They're all right in there. They can close down the what? Filtration membrane or filtration slit diaphragm, whichever one you want to think of, that's okay. They are contractile, and also they have a characteristic which is kind of unusual, but it's certainly attributed to them and been shown to be the case, is phagocytic. they seem to be phagocytic, so kind of unusual with the mesangial cells positioned there. And there we can see, them. I won't tell that, I'll tell you that next time. Anyway, that's a good point, but I'll save it later. Alright, so we have loop of Henle, and we're interested, especially here in the loop of Henle, that are the long loops of Henle, and the permeability properties of these loops. Alright, so as you look at the long loop of Henle here, descending limb and an ascending limb, okay, and now when we go down the descending limb, what happens to the saltiness, if you will, we can use that term now, I suppose, or the osmolarity, if you want, as we descend the descending limb? It increases, does it? It becomes more and more salty, or saltier, maybe a better word there, because water moves right out of the long loop of Henle there, the descending limb, but solids cannot leave. So as the fluid descends the long loop of Henle, you have now increase in saltiness or osmolarity. So it becomes higher as far as osmolarity goes up to maybe 900 or 1,000 or even 1,200 is the highest. As you make the hairpin turn, now what happens to the osmolarity? It goes down, doesn't it? And then again, we have our solutes actively being pumped out into what? Into the interstitium, okay? Into the interstitium, and now we have our fluid here that is hypoosmotic to plasma. That's always our reference, hypoosmotic. So it could be maybe 100, okay? So right now you know that in order for us to do that, concentrate urine, we need one thing for sure, which is what? Our long loops of Henle. That's what we have to have. But more specifically, we can say, yeah, we've got loops of Henle here. They are long loops of Henle, but we have to say, yes, we have long loops of Henle, and their permeability properties is what allows us to concentrate urine. Long loops of Henle and their permeability properties. Because if there weren't permeability properties here, then there would be no concentration gradient. We wouldn't be able to concentrate urine. But what is another substance we need to concentrate urine, another additional item here? We definitely need long loops of Henle and their permeability properties, but we also need, though, a hormone, which is what? Because if we continue with this 100 milliosmol fluid traversing all of our tubes here and start urinating, okay, we don't want that. We're going to rapidly dehydrate after a while, so we have to have some type of mechanism in play to say, yeah, we got 100 milliosmol fluid here, it's pretty dilute, but now we want to start to maybe reduce some of that water that's leaving. So we don't want to have this continued diuresis, right? We want to have some type of anti-diuresis going on. So what hormone is this? ADH, anti-diuretic hormone. Where does that come from again? Pituitary. It comes from the pituitary, right? And of course its target now is what? Nephron's not, kidney's not good enough, nephron's not good enough, not even is the DCT collecting duct. Now specifically we can say the principal cells, right, of the DCT and collecting duct, they have receptors on it for the ADH. What does it tell them to do? Reabsorb water, that's correct. Now we can have fluid here that will leave our collecting duct or DCT and collecting duct, which will be a higher osmolarity than 100. Reabsorb water. Okay? Actually insert aquaporins into the plasma membrane, so they reabsorb water. Okay? Trying to raise blood volume, trying to raise blood pressure. Okay? All right. So one more point about this, and then we'll move on to some other things, is that we know that if we're going to change the osmolarity of the fluid in the long loop of Henle, we rely on the permeability properties of these cells, right? If we're going to change the osmolarity of the fluid in the DCT collecting duct, how is this done? Hormonally, with hormones. One of which we named, which is ADH. What's the other one? Aldosterone. That comes from where? The adrenal gland, yes. Okay? Its target again is same thing, right? Principal cells, the DCT collecting duct, and it tells them to do what? Aldosterone. What'd you say?
Reabsorb water.
No. Reabsorb sodium, and water follows through osmosis. Okay? Reabsorb sodium, water follows through osmosis. Trying to raise blood volume, trying to raise blood pressure. Okay? Targets are the same. Okay? Different mechanisms of action there. All right? And so when we get into endocrine system a little later on, we want to figure out, which we just did a little bit right here, where it comes from, where it goes, what happens when it gets there. That's the idea with any hormone, okay? So now you have an idea of how things will progress in endocrine, but right now that's what we're interested in there. Okay, this image is up here only so you can appreciate the driving pressure, which is blood hydrostatic pressure or glomerular pressure. There are other opposing forces there, but none of them are strong enough to overcome that driving pressure right there, which is our glomerular hydrostatic pressure. That's the force that drives filtration. And we know 100% occurs in obviously the glomerulus, and then we can split up the other nephrotic processes, or at least the reabsorption. And I've talked about this already, right? We just did a bit of review with the macula densa cells there too. And this we can see here. And I explained that word paracrine, didn't I? I said it was a way of hormones, how they act on their neighbors. That's not something you get excited about, but that's how sometimes hormones are released and act. And it could have been even an autocrine type of hormone where it acts on itself, but that's maybe a confusing word if you didn't know exactly what it is. And we got to the extrinsic controls, and we were kind of right here with this, right? Renin, angiotensin, aldosterone system, is that correct? Okay, and when we were wondering, when does this system activate? When would we activate renin, angiotensin, aldosterone? When we have what? Low blood volume, yeah, and low blood pressure. Okay, this is when this system will activate. And so if we kind of look here, this is not something you have to draw out or even necessarily identify in a pathway of sorts. But you can see that we've got our angiotensinogen there, we got renin there, and there's our pulmonary capillaries with ACE, and then of course we have angiotensin II. Now if we follow this down, we've got two things identified that angiotensin II does for us. One of which goes to the hypothalamus, right? Vasoconstrictor. The other one to our blood vessels, vasoconstrict, that certainly makes sense. And then this one over here, this is another action now of angiotensin II, is to go and you can just worry about the adrenal gland. We don't have to worry about the cortex. We don't care about that right now in this chapter, so don't overthink that. But just the adrenal gland is fine. It stimulates the release of aldosterone, which will then do what? Reabsorb sodium, water follows through osmosis, trying to raise blood volume, trying to raise blood pressure. Okay? So there's another function now of angiotensin II, besides just, as you can see, brain, hypothalamus, blood vessels, and then to the adrenal gland. Okay? Release more angiotensin, or more aldosterone. Okay, and here's my laundry list of actions. But if you, and we'll go through this in just a moment, but all of them are doing this, trying to lead to what? Increase in blood pressure. So if we kind of go through here, we've got one taken care of. Stimulates resorption of sodium, which is again that action as it goes to the adrenal gland, right? Stimulates hypothalamus to release ADH. That's a new one, isn't it? Stimulates hypothalamus to release ADH. Although it makes sense, doesn't it? If we're trying to raise blood volume, another way to do it is just that, and I think I've included that before. And there's the thirst center. Constricts efferent arterioles, okay? Increasing fluid reabsorption. So if we know that the efferent arterioles are giving rise to what vessels? Efferent arterioles give rise to what capillaries? The vasa recta and the peritubular capillaries. If we reduce the efferent arteriole, okay, reduce that size, now we have an increase in reabsorptive capability, which will occur in those capillaries. Reduce this HSP, which is hydrostatic pressure. Now we've got reabsorption is promoted there, where previously it would not be. Okay, so if we reduce that pressure, now we have reabsorption occurring on that side, and there's an image of that right there. Reduce the resistance to the reabsorption, so water then flows in. Cause glomerular mesangial cells to contract. That is certainly the case as well. So really what it's trying to do is cause that filtration membrane now to go from this to that. All right, which again reduces the what? Surface area available for filtration, trying to reduce GFR, right? Reduce the amount of filtrate formed, trying to raise blood volume, trying to raise blood pressure. So you have to memorize a laundry list? Not necessarily, but you have to be aware of it's a vasoconstrictor, so let's go to the arterioles, and we're trying to raise blood pressure. Let's also think about the ways to raise blood volume too. You can add in thirst center, you can add in ADH, you can add in aldosterone, stimulation and release of that. So anything that raises blood volume and subsequent blood pressure, you can attribute, at least in your mind, to angiotensin II as doing that. So it's not a mystery what is happening here. It all does make sense, and they're all leading to an increase in blood volume, subsequently blood pressure. Okay, so could you recognize all the following actions of angiotensin II except for? And except for is going to be something that lowers blood volume and lowers blood pressure, you would think, because all of these are increasing. Okay, so kind of be aware of that. If you know a whole bunch that is doing this, and one oddball sticks out, then it's probably going to be the strange one. All right, forming concentrated urine, we talked a little bit about this already, and I've got two things listed here, countercurrent multiplier and countercurrent exchanger. So with this loop of Henle, we're always talking long loops of Henle. Why are we talking long loops of Henle here and not the short ones? Because the cortical do have loops of Henle. They are very short, or they may not have a loop at all. That's been noted to be the case as well. So why do we care that they have to be long loops of Henle? There you go. They dip down way deep into the medulla, okay? And if they don't dip down to the medulla, then they're not going to be exposed to that very highly concentrated, or if you want, very salty interstitial fluid that's in the medulla. So when these loops of Henle dip down in there, we can actually call those long loops of Henle their countercurrent multipliers. What are they multiplying? The gradient, yes. As you descend down in the medulla, we might say, let's say, 300 to 600 to 900 to 1200, it's being multiplied as you descend deeper into the medulla. That's the multiplication process there. That's why they're called the multiplier, okay? Countercurrent multiplier, okay? And we also now know that we have the blood vessels that surround our long loops of Henle within the vasa recta. They're the countercurrent exchangers, and we have an idea already that they're going to be exchanging our solutes and solution, okay? But for right now, think of, hey, you've got these vasa recta that do follow the long loops of Henle, and we're saying, they are the countercurrent exchangers, but their main job, okay, as we'll see, their main job really just to irrigate the cells deep down in the medulla, bring nutrients and get rid of waste. That really is their main job, but their orientation is such that they get this kind of moniker, the countercurrent exchanger. So their main job is to irrigate the cells deep in the medulla. But because of their arrangement, as you'll see, they get this countercurrent exchanger. So, okay, with this, I'll give you a statement here, and then I'll flip to the slide and we'll explain these, is that we can say countercurrent multiplier, or loops of Henle, they create, okay, they create this gradient that's in the medulla, okay, from 300 to 600 to 900 to 1200. They create that with their what? Permeability properties, right? Okay? But the countercurrent exchanger, they maintain the gradient. They maintain it. So these create it. This maintains it. Maintains it. And I'll ask one question, then I'll move on, is, do you think their maintenance, again, these create the gradient that's in the medulla from 300 to 600 to 900. They create that, okay, in the interstitium. But these blood vessels, or these right here, they maintain the gradient. So my question is, what was my question? Oh, yes, my question is this. Based on their arrangement, okay, what is their arrangement as you look at them as they're following the long loops of Henle? They're going down and going up, right? They're not going in a transverse fashion. So based on their arrangement, okay, they are able to maintain this gradient and not wash it out. Okay? And that's a long way to go for the location and presentation of these of this vasa recta. But they maintain the gradient because of their orientation. If they were this way across our long loops of Henle, they would wash the gradient away. Things would diffuse in like solutes, okay? And away they go, and the way they go, they'd be gone. There would be no gradient in there if these did not follow the long loops of Henle. Okay? That's important to kind of recognize from, structure governs function. The structure says they go up and down like this. So that's great, but the function now says, hey, could they do that? They just exchange solutes and solution, and they just do this passively, just by diffusion in or out. That really is not their main job. because their main job is to irrigate the cells deep down in the medulla. But because of their arrangement, they play a key role in maintaining the gradient that's there. If they went this way, away the solutes would diffuse right in, away they'd go, there'd be no gradient, there'd no way to concentrate urine. So there's kind of a neat system there in that the arrangement provides for this exchange system, okay, and not get washed away and removed, okay. All right, so we also mentioned this before ADH. Recognize abbreviations too. If you haven't recognized that as antidiuretic hormone, do, and you're welcome to use that abbreviation as well. In fact, when we get into the endocrine system, almost that entire chapter is abbreviations. So be able to recognize them and use them. All right. Osmolarity. We got that. Was 300. And this is already I've talked about here, so I don't have to say that. Sometimes I bring this on the first day of class, and I just did. So we've got a reference there: isotonic, hypo, or hypertonic. And we've already got terms for that, so I won't have to go over that again. But there's more countercurrent flow. So okay, so far, let's take an image of this and see. Oh, there's the gradient increasing 300 to 400 to 9 to 1200 as you descend deep into the medulla. And if you wanted to picture, you could picture the long loops of Henle going down into the deep area of the medulla too. So you can see the gradient. This is what's being multiplied. Is the gradient there as you go from 300 to 400, 600. And what's doing that again? It is the permeability properties of the long loop of Henle. Okay, what's maintaining it? Vasorectin. Yes. So here's our long loop of Henle. There we say our 300 making its way down here. That's the same as what you'd see in our blood plasma. Okay, as we descend right down here, this is just showing now the properties of the descending and even ascending limb of the loop of Henle. Water's leaving. We have now 600 to 900 becoming hyperosmotic. Make the turn at 1200 or so, and now these solids are actively pumped out, just like that. This one again, right, controls what? That one there. Now remember, you're probably thinking, well, what if, this is pumping out and the water's going out here, and that's all well and good, but, how come they just don't equal out? And just kind of equal out there and then no movement. This is just a snapshot of what's going on, a quick picture of what's happening. This is constantly going on. There's nothing, it's like when we maybe looked at the cardiac cycle. It's constantly going on. There's no start or stop point. This is a quick snapshot of what's going on. So things are moving constantly. So as you descend further, you pumped out, water is moving out, and as you go further down, okay, it becomes a steeper and steeper gradient, much more and more salty if you want. I don't think salty is a word, is it? Salty? I don't know. But more and more hyperosmotic as you descend, pumping out here. And again, it shows the properties here being pumped out at the interstitium, and then water moves out on this side, and then as you make your way up, we have now our fluid is hypoosmotic, but we don't want to have that all the time. Now, it always depends on the body's needs, okay? We may want to have just 100 milliosmol. It depends on the body's needs, but if we have, the release of ADH, okay, which is necessary to make concentrated urine, then that will change, okay? But there is actually one way to prevent the release of ADH and keep on pumping out 100, at least 100 milliosmoles fluid and lots of dilute type of urine. And one way to do that is alcohol. Alcohol turns off ADH, so now you have high amounts of dilute urine is produced. And anyone who's drank a lot of Rolling Rocks maybe once probably realizes that. So you constantly urinate, okay? And the urine is a large volume and very dilute. But what happens though over time? All of a sudden now you're starting to urinate, starting to dehydrate, and then maybe wake up the next morning, and how much urine do you expel or void in the morning and then after? Very little, right? Very little, and it's even highly concentrated, isn't it? Okay, even highly concentrated. So your body is starting to adapt and say, hey, we need to release some ADH. We're starting to dehydrate here, so we need to get some more ADH out to have this urine become more concentrated but low volume. Okay, it's a good thing we have that. Good thing we have ADH. And this is another picture of the gradient as you descend. I don't know why I have two, but you can picture that. Keep in mind again the permeability properties, and that is what's setting up our gradient here. Okay, very good. Next 100. of ADH, like I said a moment ago, and now we have our 100 milliosmol. Large volume of dilute urine, no ADH, and I said alcohol would do that. There's certain other disorders that could do that too, but we have at least one we can pick on. Alcohol turns off ADH, and so you might have large volume of dilute urine as a result of that. And then with ADH, maximal ADH, now we have our very highly concentrated urine right down there, and it doesn't have to be 1200, but that's our max that we can do as human. I've come across a couple of readings where some animals can do, I think, 10 or 12 or 15 the osmolarity of their blood. So it seems like it'd just be they urinate sand or something like that. But I think even our book or Salad's book or some of the book talks about some sort of mouse in a desert somewhere that can concentrate its urine up to 15 or 20 times the osmolarity of blood. So I don't know what mouse it was, but it was kind of interesting. Areas where the water is scarce, but nonetheless, that's that. Oops. So vasorectomy is a countercurrent mechanism removes reabsorbed water and solids without disturbing the medullary concentration gradient. They maintain the gradient. So as we have these blood vessels again going down, just like this, here they are, this is our vasa recta, going down like this. Okay? Now remember that they too, because of their orientation, they too are exposed to what? The gradient that's in the interstitium of the medulla. These blood vessels, these capillaries are exposed to that. So as we make our way down now, okay, now we have salt if you want, or sodium chloride moves in, water moves out, but as you descend further and further and further, now we have the opposite. Okay, now salt moves out, water moves in. As you go up into a gradient that is less and less. It's just the opposite. We're just exchanging salt or solutes and water. As you descend, one side it's salt going in, one side is solutes going out. In and out, just exchanging with the interstitium. And that really is not their primary function. That's not their job, but just in passing, as they are exposed to the gradient, you have movement, diffusion, movement of these solutes in and out of these capillaries, and they're simply exchanging. They're exchanging with the interstitium. The blood and the interstitium is exchanging back and forth. For no other reason, just because they're there and they're being exposed to it. That's what they're doing. So because of their orientation, they're being exposed to the same gradient as you dip down deep in the medulla, and you have salts, solutes if you will, diffusing in, water diffuses out, depending on, of course, our gradient here in the interstitium of the medulla, and then as you make your way up, now no system is perfect there, so now we have our blood almost back to what it was, 300 milliosmoles. And really, it's just the exchanger. It doesn't create it. It just maintains it. Because and the flip side is it doesn't wash it away because of the orientation. So creation, long loops of Henle, okay, right? And maintenance is the exchanger. Okay, does that make sense? Keep in mind this is both describing countercurrent flow as well. Countercurrent flow. And I think that might be enough nephron discussion. I think that's plenty of the nephrotic processes and things like that. Okay? So now we're at a point in this chapter where it needs to be discussed, but it doesn't have to be belabored for 2.5 days. Okay? And this is it right here. We have an interest, a brief interest, in the conduits, like the ureters, like the bladder, the urinary bladder, and then the urethra. Question?
Yeah, I was in the OR all this morning. After a hysterectomy, why would you need a bladder sling?
After hysterectomy, why would one need a bladder sling?
Yeah.
Well, I'm guessing that, and this is a guess, I don't know, but I'm guessing that when the uterus and was the ovaries too? It's like a total.
One of them.
One of them. Okay. So perhaps some ligament was taken out, like broad ligament. Broad ligament also helps to anchor the urinary bladder in place too. So that is my best guess. That is all I could offer for that.
Yeah,
But it
they
sounds
cut,
like it would be disturbed.
Yeah, they cut the ovarian ligament and they also cut the broad ligament too.
That would be
all of it.
what I would be thinking in that case. But maybe you can check on to see if I have some accurate statement on that. But that's my best guess. Okay, so urinary bladder. We have muscle in the urinary bladder? What kind of muscle would we have in the urinary bladder? It,
No, I'm thinking it's.
it would be smooth muscle. Smooth muscle, okay? And this is called a hollow organ, so we have smooth muscle. I actually have a name for the smooth muscle. This makes for nice trivia, fill in the blank. The smooth muscle in the bladder is called detrusor, the detrusor muscle, okay? Yeah, right up there. Detrusor, yes. Anyone heard of the drug called Detrol LA? It was for urinary incontinence. Yeah, so that's a suggesting association with that smooth muscle there. I don't know if it's still on the market or not, but nonetheless. That is the smooth muscle of the bladder, detrusor, okay? Is there epithelium in the bladder? There is, there is. So what kind of epithelium might you find in the urinary bladder? Transitional, yes, also known as, sometimes I give this also known as name. I know I gave you also known names. But that's like, transitional is right. The other name is urothelium, U-R-O, urothelium, okay? But transitional, because it undergoes changes when the bladder is full. They can be squamosal to flat, or they can be more columnar or even cuboidal when the urinary bladder is full. So transitional, okay? How do we know when we have to urinate? Yes, what kind of messages will be going up? Into the spinal cord, into the brain, and then we decide whether we can urinate or not. Right? We have that conscious control, all of us do, I hope, right? We don't have lost control of our bowel bladder functions, right? We all have that conscious decision to urinate. So what causes us to want to urinate? Yes, that's exactly right. There are stretch receptors in the wall of the urinary bladder that when they stretch like this, okay, impulses are sent into the spinal cord and then up to the brain so we can decide, yes, we have to urinate, but is it appropriate time? No, it's not, so hold it, okay? So, you have that conscious ability to override the stretch receptor stimulus when it's appropriate time to urinate. Now, eventually, though, you'll have to kind of succumb to those urges, but we have a pretty good way of overriding those urges, okay? Stretch receptors, all right? Stretch receptors, as the bladder fills up, it sends impulses into the spinal cord, and then, of course, up to, and there's our muscle here, up to the brain so that we can decide whether it's appropriate or not, right? Urinary bladder. And we know of these already, not to spend a lot of time on these structures, and they're all fine and dandy, but those are ureters. They're just simply, conduits that transport our urine from our kidney into our urinary bladder. And how does it get down there? How does it get from there to there? What's it?
Isn't it great? Precious great.
No. How does it get down there? This might give you an idea. There's a cross section of it. Yeah, smooth muscle. See, that's a cross section of the ureter, which typically has that kind of star shape. Smooth muscle. Smooth muscle causes our urine to make its way down from the urinary, from the kidneys down to the urinary bladder. Okay. Sometimes, I got a question. How do kidney stones do? Good question. I'll talk about the kidney stones in lab and what they're made of and all that. But a lot of times they lodge right in there. They can lodge right. They don't have to. They could lodge up in here too, right in that whole renal pelvis area. They can be so big that they can become what's known as a staghorn. This is extra. A staghorn calculus. Huge, so that it takes up the whole area right here. This whole area in the renal pelvis is completely filled up with a kidney stone. Huge. It destroys the kidney is what it does. But those are big. Those are really big. But sometimes kidney stones do get lodged right in here, and that's very uncomfortable, isn't it? Very uncomfortable. Because kidney stones aren't like BBs where they're smooth all around. They're what? Jagged, yeah. So as it makes its way through this epithelium here, get your attention. It gets your attention because this is not a pleasant area, all right? And of course, as it sits in here and makes its way out the urethra, also very unpleasant. Because these aren't BBs, these are rough edged, jagged structures that can really irritate the epithelium and even cause bleeding too. They can do that. So that's one reason why you might find blood in the urine, which we could call what? Hematuria. Hematuria. Blood in the urine. Okay? Blood in the urine. That's one reason why we could have it. Or I could punch in the kidney. That could do it too. But, yeah. Hematuria. Alright. I was on the agenda for today. Alright, hematuria. Blood in the urine. there's lots of causes for that, but that's one. A kidney stone certainly can cause that. And I'll talk more about this in the urinary lab as well. Anyway, any questions about transitional detrusor, how we urinate, right? We have receptors here, stretch receptors. The picture is only so you can kind of appreciate the fact that yes, we do have connections here that are going to be evaluated in the spinal cord up to the brain, and then of course we can contract our muscles here, the external one anyway, so that we can urinate. We probably realize that we don't have any control over the contraction of the detrusor because that's smooth muscle, but we can decide when and how to urinate, and that's where the conscious control comes into play, where we can say, yes, I have to urinate, but I'm not going to do it yet, so this sphincter right here, what's a sphincter? That's all it is, a circular muscle, yep. That's it. We can relax that urinary sphincter and then let the urine flow out of the urethra, okay? Stretch receptors, that's what I want you to take away from that, okay? And we need a couple words now for urination. We had voiding, right? Micturition. Micturition is another word for that. And that might be about it, I think. Urination, micturition, and voiding. Anybody know what this is talking about? Parasympathetic fibers of the pelvic nerve. What are those fibers called? Those nerves had a special, and this is actually, then we'll get on to new stuff. Remember the pelvic splanchnic nerves? Anybody remember those? S2 to S4? And there's S2 to S4 right there, pelvic splanchnic nerves. Goes down to stuff in the pelvis, right? You haven't. If you don't know that, that's okay. I just was pointing out for those who do. That takes care of urinary system. That's enough urinary system to be dangerous, I think. So that is plenty. Although, do remember in lab, when we get to lab, not tomorrow, but the next time, is that we will be doing UAs, urinalysis, okay? And so try not to urinate until a week. Anyway, right? Yeah. We'll be doing UAs, and of course you have to also not just do that, but we'll also be doing the models that we have to look at as well. Because we have models of nephrons, I have models of kidney, and we have to look at all these parts. And of course, you already have the list of all the structures that you're responsible for, at least for lecture lab exam number 3, right? You should have that on campus, all of those. Okay, so look at the renal portion, you'll see a list, a laundry list of about 10 or 20 or 30 items or so that we have to look at.
So that'd be our last lab exam, right?
It will be the last lab exam. So we've got respiratory, which we're going to talk about already for tomorrow, and then we'll have urinary, digestive, and reproductive. Okay, that's what we'll have on the last lab practical. All the anatomical structures. At no time will it be explain this or explain that. It will be name that structure. The stickers on stuff, you tell me what it is. Like I were to put a sticker on the vasa recta, name those structures. Vasa recta. And you can see all those. I've got great models of those and plenty to go around. I think I got 4 or 5 models of the nephron to see. So great stuff to see. So we'll take advantage of that another day. All right, so let's talk a little bit about digestive system. We have a couple days for this. I think I now have 2.5 days now with digestive since we've got some playtime now. So let's talk a little bit about digestive system here. And when you think about the digestive system, it doesn't build things up, it breaks things down. More of a catabolic system, okay? Rather than an anabolic system building things up, it breaks things down, catabolic system, right? And when you also think of digestive system, we can list, and I will list all these anatomical structures too that we have to mention, but also with digestion as a whole, it comes in 2 varieties. One is mechanical digestion, okay? Breaking big things down into small things. Mechanical digestion, big things into small things. And the other one is what? Anybody guess? And it can be examples, so you can make a pretty good educated guess here. If we think of mechanical digestion, big things into small, think about the teeth breaking up big pieces of something into small pieces of something. So that's mechanical. What would be the other type of digestion? It is chemical. In this case, it involves what? Enzymes. It involves enzymes. Okay? We'll come across lots of enzymes that we can identify and even give special names to based on where they're coming from or what part of the oral cavity, or what part of the GI tract, gastrointestinal tract, that they're associated with. Like for instance, if we're looking at salivary glands, we might say a salivary enzyme. Okay? okay? Or if we're maybe in the GI tract, maybe the duodenum or something, we might just have an enzyme down there. It might be associated with the duodenum or the pancreas or something like that. We can name these enzymes based on what structure we're looking at, okay? Enzymes, this is chemical digestion. Always think of that. When you think of chemical, think enzymes, right? When you think of mechanical, think of big something broken down physically into small something, whatever it may be, okay? And so if we kind of look at our conduits, our pathway here, we have lots of things to recognize, and really as humans, we're a tube within a tube. That's what we are, a tube within a tube, okay? So we got to identify the inner tube, right? And we want to keep the things that are on the inner tube inside. We don't want anything on the inner tube getting out into the outer tube, because that would be bad. Why would that be bad? What if maybe this thing opened up? You'd end up with extreme pain. Yes. You end up with peritonitis. You could, if the stuff in this tube out you see here comes out into this tube, which is in here. Okay, you don't want that. So my point is, we're a tube within a tube, and all this is the tube you see, and this is the outer tube right here. Okay, that's what I mean by that, and it's a long way to go for peritonitis, but nonetheless, we want to keep the stuff that's in this tube in that tube. Okay, so let's start at the top here, the oral cavity, and we can identify some things here, like we've got the tongue there as well, and the tongue actually does play a role in digestion. How come? How do you think it does? Or what? The digestive system, I should say. Let me be clear so I don't mislead anyone. Break up? Yeah, I'll buy that, but you're right. But what about? Was that? Pudding, okay, yes. It can move it around. It moves it around, right? When you go with that, it moves the food around, okay? But it also, though, plays a role in what we're going to eat. How come?
Taste buds.
Taste buds. We find taste buds associated with the tongue. So it does play a role in the digestive system, because if we don't want to eat it, then we're not going to digest it if we don't like it. So this is a role that the tongue plays, in addition to moving food around in the oral cavity. Okay, taste buds. We'll have to look at those too. So as we kind of go further in the posterior aspect of the oral cavity, we see the parotid glands, sublingual, and submandibular. These are all salivary glands. I know I showed these glands to some of you that went to the cadaver lab. Some of you didn't. So you didn't get the 10 points, you know. I'm kidding. Right. But anyway, I showed you these glands, the submandibular, and I even showed the parotid gland as well. The sublingual I didn't show. Those are difficult to see. You actually have to take up the floor of the mouth to see those. But you can see the epithelium that covers them. If you lift up your tongue and look at the floor of the mouth, you'll see some V-shaped structures underneath your tongue like this, okay? If you stripped off that epithelium, you would see those sublingual glands. So look at the V-shaped arrangement. Those are sublingual glands. In fact, the ducts. Why does it have to be a duct? Because it's a exocrine gland, isn't it? The ducts actually empty right on the surface of those V-shaped structures, but the glands are underneath. Okay? So take a look and see those V-shaped things. Those are sublingual. These are all salivary glands, and they produce what? Saliva, right? What's in saliva that helps us digest stuff? Now this is going to be chemical, right? Yes, we have something known as salivary amylase. This is anytime you hear the word ASE, ACE is an enzyme, salivary amylase. This is an enzyme that we find in saliva, salivary amylase. Okay? And so we also find in here, I heard lysozyme, and immunoglobulin A was in there as well. These are good to know for maybe another chapter. You're right, but let's circle that one there, salivary amylase. This is one associated with digestion, not so much immune system, but you're absolutely correct. Won't take that away from you there. And salivary amylase. This is an enzyme that acts on, what does it act on? Before you answer, think about the things that we eat. What are our foodstuffs that we eat? That's not actually a made-up word. It sounds like a silly word, foodstuffs, but it really is a real word. It's strange. What are the foodstuffs we eat for energy?
Carbs.
Yeah, carbohydrates, proteins, and fats. Okay? So our choices, and we could put water there too if we wanted, right? Okay? What do you think amylase acts on? 33 to 30% chance of getting it right.
Carbohydrates.
Yes, it acts on carbohydrates. okay? Salivary amylase. So if you don't believe me, take a cracker, okay? Put it in your mouth, suck on the cracker. What is it in 10 seconds? Gone, right? Gone, yeah, for the most part, it's gone, okay? So it works on carbohydrates. What if you took a peanut, sucked on a peanut for 3 minutes? What is it after 3 minutes? Still a peanut, right? So does that sound like that we do not have an enzyme in our saliva that acts on proteins? That is correct. That is correct, okay? Now take a piece of fat, suck on a piece of fat for 3 minutes. So what is it after 3 minutes? Still a piece of fat. However, a little kind of asterisk here is we do have some salivary lipase, a little bit of salivary lipase that's in our saliva. It's not very effective, and even sometimes we can call it lingual lipase. It doesn't matter; those terms are interchangeable. Lingual or salivary lipase, and lipase acts on fats. It's not very effective. It really is not, but we do have it. Even we'll find out in the stomach, we have a lipase down there known as gastric lipase. It's a bit more effective than this, okay? A bit more effective, but not overly still, okay? But we do have some of that, but that not enough to really make a dent. So with this, so far we've kind of got a couple of enzymes identified here in the saliva, and then you say, you know, what's the pH of saliva? Yeah, it's mildly acidic, not like stomach acid, but mildly acidic. Even it can range to a bit basic or so. So we can like 6.8 to maybe 7.4 or so. That's a good range. That's not a range I'm going to hold you to, okay? But kind of keep in mind there's a range. My point here is this: these enzymes, at least the amylase anyway, is pH sensitive. It only works in that pH. Does that make sense? Only works pH, because if it gets down to the stomach, what's the pH of the stomach approximately? Anybody know? 1 to 2, because of the presence of HCl, hydrochloric acid, okay? So, what happens when this gets down in the stomach? It gets quickly denatured, destroyed, okay? pH sensitive, okay? So does it sound like maybe that once we kind of get to different areas of our alimentary canal, which is a big long word to say this entire tube, right? Alimentary canal, that we have to include other substances here that can survive in the pHs that are present and even ever changing throughout this alimentary canal. And that's a case with this. This is pH sensitive. It gets down into the stomach, denatured, and it quits working. Okay? Provided that it's exposed to the gastric juice. If it's wrapped up inside of the bolus, B-O-L-U-S is this kind of big mass of food that we swallow, kind of a weird term there too. But bolus, if it's inside the bolus, no, it won't be exposed to the gastric juice down there. But and then it will work inside the mass of food. But if we're on the outside, it will be quickly denatured. So just kind of be aware of that. Yeah, if it's inside, it won't break down. But if it's exposed to the gastric juice, which is a fancy word for stuff in the stomach, pH about 1 or 2, quickly denatured. Okay, so we've got now our big bolus of food. We've got it lubricated with the saliva. We decide to eat it because the taste buds say, hey, we like this salty stuff, so we eat it, right? And we'll get to the taste buds a little bit later. And now we start to initiate our swallowing, okay? And we're going to the esophagus. Do we initiate swallowing, or does swallowing just happen on its own? We do initiate it. So what kind of muscle would be a large percentage of the esophagus in the upper region anyway? It is skeletal muscle. In fact, we have muscles known as, you can just sit and listen here. You don't have to write this down. It's a nice way of just hearing things as a collective presentation here. But we have muscles that are stacked on top of each other, like kind of the drinking cups, like the red solo cup kind of thing. As they stack like that, they're actually called constrictor muscles. You might be interested to know, and then go superior, middle, and inferior. And as they constrict, right, they're squeezing our bolus of food down the esophagus. Okay, constrictor muscles, superior, middle, and inferior. They squeeze this bolus of food down the esophagus. When it gets to about the middle 1/3 or so of the esophagus, we quickly have a change in muscle. It becomes a mix of skeletal and smooth. Okay, But then as you make your way down to the distal inferior portion here of the esophagus, inferior, not the distal, but the inferior portion of it, it becomes exclusively smooth. We lose control of the movement once it gets down in there. So our smooth muscle then takes over right in there in the inferior aspect of that esophagus, smooth muscle. Right? What do you think goes on in the esophagus? Anything? Any digestion, any absorption, anything at all? Nothing goes on in there. The only thing that I can say that goes on in there is lubrication. There are glands in that produce a mucus that allow our bolus of food to make its way down with much greater ease than if we didn't have the glands there. Okay? There is no digestion, there is no absorption, and what does that word mean?
Taking things in from the outside.
There you go. Taking things in from the outside that have not been in the inside before. Absorption, that's what we do in the GI tract, okay? And once it enters into the blood, it's been absorbed. Okay? So, there's no absorption, no digestion in the esophagus. What about the oral cavity? Is there any absorption in there? We know digestion is going to be chemical, but is any absorption in there? And the one that comes to mind, and some of you in clinical orientation or something might have more detail on this, but there is one, any absorption in the oral cavity? Yes, because stuff that needs to be administered in the oral cavity is positioned under the tongue or sublingual. The epithelium there is much thinner than any other place. So you can have absorption in the oral cavity, like nitroglycerin is one of those. Nitroglycerin converted into NO, and quickly is a vasodilator, right? Good for heart problems, right? Circulatory problems. NO, okay, nitroglycerin, okay? A potent vasodilator. This is one thing that can be absorbed in the oral cavity very, very quickly. The epithelium under the tongue is very, very thin. Okay? Alright, so we've got some absorption going on, at least now, and some digestion going on in the oral cavity. We've talked a little bit about nothing happening in the esophagus, right? And we'll come back to mechanical digestion in the oral cavity as well, but that's not a huge ticket item, but it's still something to be addressed. We follow down the esophagus and go through what opening in the diaphragm? Esophageal hiatus, very good. We enter into the stomach, okay? And when we're in the stomach, we have this big bolus or mass of food right in here, and it goes right into the stomach there, and it exposes immediately to the pH of the stomach, which is about 1 to 2. So very, very acidic in there as well. What does the HCl do for us? At least now, as you know it now, we'll add a few other things too, but what does HCl do for us?
Breaks down.
What's that?
Breaks down.
Yes, it does break down proteins, absolutely. It also destroys things that we eat, like pathogens. We eat a lot of those, right? It can destroy those very, very quickly. Nothing can live in that pH of 1. Very, very acidic area. Gastric juice. Notice I'm saying gastric juice, so that probably implies that it's not just HCl. Other substances are in there too. We'll explore what they are as we go on. What do you think the job of the stomach is? And does any absorption take place in there? Let's do the first one first. First one first? Sure, that's a good one. Let's do that. What's the job of the stomach as a whole? Any ideas?
Chemical digestion.
What is that?
Chemical.
Chemical digestion? Not primarily. Not primarily. Its main job is to function as a holding tank. Holding tank. Okay, it functions as a holding tank, right? And it also has the capacity, and you'll be able to recognize this when we take our cats back out of the bag in the digestive lab. We'll open up the stomach in the cat, and we'll be able to see the folding of the mucosa in the stomach called rugae, all right? And now, provided your cat didn't eat. If your cat did eat, your rugae, as you dig out maybe the dead mouse or something like that, pull it out, okay, your rugae will be all flattened out, you won't see them, okay? So the rugae allows the stomach to unfold upon itself like that. We can all experience that probably in the third Thursday in November or something, or maybe the 4th, whatever it is, okay? You can test that theory if you want. But that's what the rugae allows for, is to unfold upon itself and fill up and function as a holding tank. But now we've got food in there, right? But it also doesn't just hold it and sit it like this, okay? It also does what? You said earlier. Churning, okay? We have muscle in there, right? being a hollow organ, it's going to have smooth muscle. But it's got muscle that's oriented in a circular fashion, it's got muscle that's oriented in a longitudinal fashion, and a transverse fashion. So we have food in the stomach, it can do this. Churning, churning, churning. What's it mixing? Bolus of food with stomach acid, and or collectively now we can call it what? Gastric juice, okay, which is one of the components of stomach acid. So it's mixing, it's churning, it's mixing and churning, okay? And when it becomes liquefied, okay, whatever's in there, maybe it ate a mouse herself, I don't know. When it mixes up like this, okay, and it becomes liquefied, then we actually change names. It's no longer just bolus of food with gastric juice, it becomes chyme, C-H-Y-M-E, chyme. Is this liquefied stuff now that's in the stomach, chyme. So chyme now is formed in the stomach. What's the pH of chyme? And I'm not asking just so we can have a laundry list of pHs. We have to figure out this is the pH of this, and as it goes through the alimentary canal, we have to change the pH, because some enzymes don't work in certain pHs, as I said. So what is the pH of the chyme? About 1 to 2, right? About 1 to 2. Okay, very good. So we get an idea so far. This is just an idea of what's happening here. So maybe you're kind of getting the idea too, with a little bit of detail sprinkled in. All right. So now as we're in the stomach here, but you also can notice we have organs here that aren't part of the alimentary canal. That means they're not part of the tube. Okay, this is the alimentary canal, the big tube here. We have organs that we can designate. There's our liver right there, the right upper quadrant, and then the gallbladder tucked underneath. We can call that, in addition to the one hiding posterior to the stomach, the pancreas, we can call those organs accessory organs, okay, of digestion. They're not part of the alimentary canal, they're not part of the tube, they're accessory organs of digestion. They play major roles in digestion, okay? In fact, maybe someone remembers from A&P 1, when you learned about that organ, okay, because we had to identify organs that belong in the digestive system and organs that belong in the endocrine system, right? You should remember that. If you don't, now would be a good time, because you remember the pancreas saying, ooh, the pancreas is an endocrine organ, and it's also digestive, but now we're in digestive right now, would you think that the pancreas is a major digestive or a major endocrine organ? It is a major digestive organ. In fact, 99% of the pancreas is dedicated to digestion. Only 1% is dedicated to endocrine. Okay, 99% is digestive, and only 1% is endocrine. Pretty amazing there. So again, these are accessory organs of digestion here that I'm mentioning. I mentioned the pancreas because I think that maybe you should recall that. I would think. Okay, that's the case. So now if we leave the stomach. Okay, we leave the stomach, we go into the first portion of the small intestine that is known as the duodenum, and there we can see the duodenum right here. And I showed some of you this on the cadaver, the duodenum, and just as a kind of point of interest is that it's C-shaped like this. The duodenum looks like this, even with a little hook on, as a matter of fact. Not because my thumb's funny, but because it goes like that. There is a hook, and in that C-shaped concavity sits the head of this organ. The pancreas sits right in there. Okay? So bear that in mind as you're learning about the relationship between the pancreas and the duodenum. They are intimately related right there. So we leave the duodenum, okay? Even though we have to discuss what happens, we're just going through now our alimentary canal, okay? Lots of good things happen in the duodenum, okay? We leave the duodenum, we go into the second portion, which we can't hardly see here, but now we have the jejunum. The jejunum is the middle portion of it, jejunum, okay? And the duodenum, I guess we don't even have to figure out why they're called that. We don't care. I like this stuff. But jejunum meant empty. That's extra. It means empty. Whoever decided to open up the jejunum on the cadaver many years ago, okay, opened up the jejunum on many cadavers and said, hey, this is empty. We're going to call it the jejunum. It's empty. Okay? Jejunum. All right? So duodenum, jejunum, and then we leave the jejunum and we go to the ileum, which is the last portion of the small intestine. You've heard that word before with regards to the lymphatic immune system, right? The ileum. What was the big deal about the ileum? You found what? Peyer's patches, right? Peyer's patches were in the ileum. Okay. All right. That's where you found Peyer's patches, the distal ileum. Okay. Clusters of B and T cells, right? So we found them there. So we leave the ileum, and then where do we go? Small intestine we've got done. Now we go into the large intestine, okay? We can call large intestine the colon if you want, but we don't have a small colon. All right, there's no such thing as small colon. There's small intestine, or there's colon, or large intestine, but no small colon. So just as kind of a point of interest, is that. There's a colon, large intestine, and the ileum, okay, dumps right into the first portion of the large intestine, known as the cecum, and that actually means, it doesn't matter what it means. It doesn't. It's fun to do. But anyway, it dumps right into the cecum, okay? And so as you can see, maybe you can't, but ileum dumps into there. There's actually a little valve there that regulates the release of the ileum contents into the cecum called the ileocecal valve. Ileocecal valve, okay? We'll find a valve there. There are other valves positioned along here that I didn't bother to name, but I will as we go.